Okay. Okay, perfect. So today we're going to be discussing both cardiac and smooth muscle physiology, right? So quick disclaimer before we begin, I do tend to speak a bit quickly. So if at any point you guys need me to re-explain a concept, or if you need me to go over something again, just let me know, and I'll be able to do that. I'm following you guys through the chat, and you can unmute and just speak at any time. And with that, let's begin. So very quickly, back to the basics. We have three types of muscles in our body. We have the skeletal muscle, the smooth muscle, and the cardiac muscle. If you were to separate them into two groups, you would do so into striated muscles and non-striated muscles. Striated muscles are your skeletal and cardiac muscles, while your non-striated muscles are your smooth muscles. Why do we separate them into those two groups? This is because the striated muscles have a structure called the sarcomere. The sarcomere is just basically a very specific arrangement for the contractile apparatus of your muscles. So both your smooth, skeletal, and cardiac muscles, all of them have this contractile apparatus, but only your skeletal muscles and your cardiac muscles have them arranged into sarcomeres. And the sarcomere is what gives this histological appearance of dark, as you can see here, like a dark band and then a light band. On the other hand, your smooth muscles do not have a sarcomere, but they're still able to contract, okay? They still have a contractile apparatus, they just don't have a sarcomere. Now let's begin with the actual lecture. What is a cardiac myocyte? Myocyte just means muscle cell. So they're contractile cells that will contain striated fibers organized into a sarcomere. And we'll get into details about a sarcomere in a minute. Second of all, they're branched. What does that mean? If you guys take a look at this diagram over here, you can see how one singular cell is literally branching. So we're just talking about the shape. Not all cells do this, only cardiac cells do this. Second thing is that they also have a single nucleus. Some cells have more than one nuclei. This is not true for cardiac cells. They only have one single nucleus. Finally, they are connected to each other through intercalated discs, which can be found over here. Okay, what are intercalated discs? Think of them as mini gateways between the cells because you're trying to allow the electric signals and you're trying to allow all these ions to spread throughout the cells, right? So you need those intercalated discs to act as doorways between the cells. Okay, very easy, very simple. Now we said sarcomere like at least 3 times since the beginning of the lecture. What on earth is a sarcomere? A sarcomere is the basic functional unit of a muscle fiber. This is the textbook definition, and you do need to be familiar with it in case it comes in an MCQ. It's the basic functional unit of a muscle fiber, and it's responsible for muscle contraction. Now, when it comes down to the structure-wise, they have both thick and thin filaments. So let's take a closer look at this. Your thick filament is what's known as a myosin molecule or a myosin chain, whereas your thin filament is your actin or light chain, okay? So once again, you have your thick filament, which is your myosin molecule, and your thin filament, which is your actin chain. The myosin molecule will contain the myosin tail, and then it's going to contain 2 myosin heads. And something that will come up again over and over again in this lecture is the fact that the myosin heads have myosin ATPase activity. So I really need you to remember this. This is going to come up again at a later stage, and I'll explain it in further details then. On the other hand, your actin has your actin molecules over here, those circles, and then it has a tropomyosin, and then it can have troponin depending on what cell you're looking at. So if you're looking at a smooth muscle cell, you will not have troponin. But since you're looking at cardiac cells right now, you will have troponin. If this is confusing, that's fine. We will go over this again later on through the lecture. Okay? So for now, what I want you to remember, thick filament, myosin, thin filament, actin. Let's move on. So the way your cardiac cells are arranged is that over here, you have this entire cardiac cell, okay? This is one cell, this whole diagram. And then those cells are made up of a bunch of tinier structures called a myofibril. And then this myofibril is made up of sarcomeres that are arranged in series. Basically, they're arranged back to back. So if you guys remember from physics, you took that when you're arranging an electric circuit in series, it basically looks like this, right? So that's the same concept. You're arranging the sarcomere in series, you're arranging them back to back. So that's exactly what this slide says. A single muscle cell has several myofibrils, and the myofibrils is made up of sarcomeres that are arranged back to back. And over here, I just put like one singular sarcomere. This is what a one singular sarcomere looks like. And as I said, your sarcomere is what gives you that dark and light appearance in histology. So the dark band is made up of the A band, and then the A band is going to have something called the H zone, which is like a lighter colored region within the dark band, okay? And then the H zone is going to have an M line coming right down in the middle. So let's take a look at it over here. This is the A band. This whole thing is the A band. and then you have the H zone, and then you have the M line. It may seem confusing, but I think you guys have a histology lecture on this. And also, there's not much to it. You just have to kind of memorize the structure. So remember, A band is the bigger region, and then inside of it, you have the H zone, and then in the middle, you have the M line. On the other hand, for your light band over here, you'll have the I band, that's the light band. And then in the middle of the I band, you'll have the Z disc. Again, there's nothing more to it here. You're just memorizing, honestly. One last thing before we move on is remember how we said the sarcomeres are arranged in series? So when they're shortening, they're going to shorten in on each other like this. And that's just going to cause the muscle to like shorten. So over here is how it looks like when it's relaxed. This is the full length when it's relaxed. And then when the muscle is shortening in series, because they're arranged in series, they're arranged back to back, it's going to shorten like this. That's all the slide is saying, nothing more to it. So very quick checkpoint, very easy question. Who can just type in the answer for me? Can you guys just hear? Can you guys hear me clearly? Okay. I did get confirmation that you guys can hear me clearly, but since no one's answering, I'm just gonna assume that you guys are too tired because you just attended another lecture. That's fine. But if anyone okay, thank you. It is sarcomere. It is B. Thank you. Moving on. We're going to talk about the electrophysiology of the cardiomyocytes. Oh, apology. Okay, my bad. Thank you so much for bringing that to my attention. The answer is C and not B. I'm sorry. So the answer should be C, sarcomere. Thank you so much, Ibrahim. Moving on, the electrophysiology of the cardiomyocytes. So we're very briefly going to touch upon the electrophysiology of the heart. So the heart is special because it has a special group of cells called pacemaker cells. Now, what does that mean? Pacemaker cells have a special feature called autorhythmicity, or the ability to depolarize on their own. So usually depolarization in cells occurs because of a prior signal. It could be because of a stretch. It could be because of some form of receptor being activated. But these pacemaker cells are special is because they do not need that. On their own, they can start to spontaneously depolarize. And we have two groups of pacemaker cells. We have your sinoatrial node cells, and then you have your AV node cells, okay? Your sinoatrial node cells are your primary pacemakers, primary pacemaker cells. So they set the heart rate for your heart, and your heart will follow that heart rate. Okay, let's just go through the bullet points one by one. So sinoatrial node cells are autorhythmic cells. We already explained what that means. They depolarize spontaneously. They generate the signal for the myocardial contraction. So once they depolarize, that signal will spread throughout the heart, and it's going to tell the cardiomyocytes to contract right now. They do not have sarcomeres. That's because pacemaker cells are not contractile cells. Okay, keep that in mind. Pacemaker cells are cells that create electric signals. They do not contract themselves. And sinoatrial nodes specifically will set the pace of the heart at 70 to 86 beats per minute. This is what a normal heart rate is supposed to look like. So in a normal human being, that's your normal heart rate. And it's set by the sinoatrial node. But you do have another group of pacemaker cells called the AV node cells. And they do have a different heartbeat. They do have a different heart rate, which is 50 beats per minute. So you might ask yourself, why do we follow the sinoatrial node's heartbeats then? It's because it is the primary pacemaker cell. So unless you have damage to your sinoatrial node, or there's some form of electric conductivity issue, then you will continue following the sinoatrial node. If this is damaged, then you will begin following the AV node, and that's when the patient begins having issues. Furthermore, you have Purkinje fibers. Purkinje fibers they're not really pacemaker cells, but in the case of damage to your other two pacemaker cells, they can act as pacemaker cells, and their heart rate is 25 to 40 beats per minute. I would suggest that you guys memorize these numbers because he did highlight them in his PowerPoint. So now let's move on to how the electric signal will actually spread throughout the heart, okay? We said you have your primary pacemaker, which is the SA node. That is the primary pacemaker cell. And it's going to spontaneously depolarize and send electric signals to the rest of the heart. So how does it do that exactly? It's going to do so through the internodal pathway. So first of all, the sinoatrial node will depolarize spontaneously and send an electric signal. This electric signal is going to go through the internodal pathway, and it's going to cross the atria. Once it crosses the atria, it's going to reach something called the AV node, which we just discussed. Now, what's going to happen at the AV node is that it's actually going to slow down the conduction of these signals. Why? Because once you guys have the electric signal, contraction is going to follow quickly. And you don't want your ventricles to contract just yet because you want to allow them to relax and fill up with blood. So let's just say that one more time. Your sinoatrial node will depolarize. It's going to send the signal to the internodal pathway, which is going to reach the AV node. Once it reaches the AV node, it needs to slow down the signal a bit. It's not going to stop it. You're not stopping the signal. You're slowing it down. Why is it slowing it down? Because you want to allow your ventricles time to relax and to fill up with blood. Then the signal is going to go down the AV bundle. AV bundle stands for atrioventricular bundle because it's between the atria and the ventricles over here. And then it's going to go down the bundle branches. And then it's going to go down the Purkinje fibers. Once it reaches the Purkinje fibers over here, the signals are going to move to the cardiomyocytes, and they're going to cause depolarization from the apex. This is the apex of the heart, all the way across the base and back up. Is that clear? Do we have any questions? Okay, I'm assuming there's no questions. So who can quickly answer this question for me? Okay, once again, since I'm not getting any form of response, as long as you guys don't have questions, I'm fine with moving on with the questions on my own. Okay, thank you, Lean. Is it B? Yes, absolutely. It is B. It is the AV node. So if you guys got confused, what do we mean by relay station? We mean at what point will the heart rate slow down in the ventricle? I mean in the electric conduction pathway of the heart. Okay, and it is the AV node because, as we said, it needs to slow down at the AV node specifically to allow the ventricles time to relax and to fill up with blood. So I hope that cleared any questions up for you guys. Now we're going to be moving on to a more tricky part of the lecture, so I would appreciate if you guys just focused with me and paid attention. We're going to be discussing the action potential of the heart. And this really depends on your understanding of the normal action potential. So if you guys have not studied this lecture before, I would recommend studying the normal action potential lecture first before approaching this lecture. And we will start at phase 4. So under the normal conditions, your cardiac myocyte, let me just write that here, your muscle cell, exists at phase 4 normally if there's no depolarization and no contraction happening. Okay, so you normally exist at phase 4, and this is mainly because of the potassium ion. So what determines the resting membrane potential of the heart? It's the potassium ion. Just memorize that and really be like sure of it. Okay, and what's going to happen is, as we said, the sinoatrial node is going to send an electric signal for your cardiac cells to contract, and that is going to cause depolarization. So you're going to depolarize, and your positive charge is going to increase until you reach negative 55 millivolts. Once you reach negative 55 millivolts, this is going to cause the voltage-gated sodium channels to open up. Okay, so let's just say that one more time. Phase 4 is your normal resting membrane potential, and this is determined by your potassium ions. Then your sinoatrial node depolarizes and sends signals all the way down that pathway that we just discussed, and it's going to tell your muscle cells that it's time to contract. This is going to cause depolarization until you reach the negative 55 millivolt level, and then that's going to cause the voltage-gated sodium channels to open up. Once the voltage-gated sodium channels open up, you're going to have the huge influx of sodium into the cell, okay, as seen over here. So if you guys are confused why the sodium entered the cell, it's because the amount of sodium outside is much more than the amount of sodium inside the cell. So it's going to move down its concentration gradient, and it's going to flood inside the cell, causing a very massive upstroke. So if they ever ask you in an MCQ, what causes the massive upstroke in the action potential for the heart, you are supposed to say sodium. If they ask you what causes depolarization of the heart, once again, you are supposed to say sodium ions. I hope that is clear. Now, another thing that you need to keep in mind is that these channels are going to close on their own within milliseconds, okay? These sodium channels are going to inactivate on their own within milliseconds. So this process is very quick, and this is why we call it like a massive upstroke. Now, once you guys reach all the way around here, like they didn't specify, you should know the negative 55 millivolt value. So you should memorize this. But they didn't specify any other values. So just understand that, okay? Once you reach around somewhere between +10 and +30 millivolts, what's going to happen is that your transient outward potassium channels are going to open up. So you reach somewhere between 10 to 30 millivolts, and your sodium channels will close, but your potassium channels will open. And when your potassium channels open, what's going to happen is that your potassium ions are going to leave the cell. Once again, why does that happen? This is something that you do need to memorize. The potassium concentration inside the cell is much higher than the potassium concentration outside the cell. So what's going to happen is that the potassium will flood outside. Is that clear to everyone? Does anyone have any questions up until now? Okay, I'm assuming not, since no one's interrupting. Can you recap the potassium part? Absolutely. So once again, let's just clear the slide very quick. We said you're at phase 4, you're normal. Then you get a signal to depolarize, and that causes the sodium channel to open. Is phase 4, wait, I'll answer your question, Reena, in a minute. So you're at phase 4 is normal, and then you get a signal to depolarize. That is why the sodium channels will open at phase 0. And then once you reach between 10 to 30 millivolts, once you reach this area, your transient outward potassium channels are going to open, okay? And that's going to cause potassium to leave the cell. Why does potassium leave the cell? Because the concentration of potassium inside the cell is much higher than the concentration of potassium outside the cell. And you know, your ions will always diffuse from an area of high concentration to an area of low concentration, okay? So I hope that explained it. You're welcome. Now, Leon, is phase 0 where contraction happens? No, phase 0 is not where contraction happens, not yet. We'll get to the phase where contraction happens in a bit. Phase 0 is just the depolarization phase. Now let's move on. So we did phase 4, 0, and 1. Now we're at phase 2. So let's just recap real quick. We said at phase 1, you have the opening of the transient outward potassium channels. This causes potassium to leave the cell. But what happens is that normal action potentials look somewhat like this, okay? They just depolarize and then repolarize instantly. For a cardiac myocyte, you do not want it to depolarize and repolarize instantly, because you want some time for the full contraction to occur, okay? Because if you just have contractions like this, then the contractions are going to be incomplete and they're going to be weak. So what you want your action potential to look like is that you want it to look somewhat like this, okay? You want something called a plateau. You want it to last longer to allow a full contraction to happen. This is why the plateau phase exists. And how does this plateau phase exist? Like, what makes it happen? Basically, your transient outward potassium channels, or just for short, your potassium channels are still open, and they're still causing potassium ions to leave the cell. This is going to decrease the positive charge inside the cell because potassium is positive, and if you have positive charges leaving the cell, then that's going to decrease the positive charge inside the cell. At the very same time, somewhere around, let's say 5 millivolts, your L-type calcium channels are going to open, okay? So, and this is where phase 2 begins. So you're going to decrease your positivity in the cell until you reach around 5 millivolts. So we went from 30 to 10 down to 5. And then at some point, the L-type calcium channels are going to open up. Once they open up, the calcium is going to start flooding into the cell. Again, why does that happen? Because the calcium outside the cell is much higher than the calcium inside the cell. So once you open this tiny gate, they're going to start flooding in, okay? And since you have calcium entering the cell and potassium leaving the cell, and they're both positive, what's going to happen is that they're going to equal each other at some point. So the amount of potassium leaving the cell is the same amount as the amount of calcium entering the cell. And that is what creates our plateau phase. I feel like this part is kind of tricky. So is that clear to everyone, or should I repeat? Okay, great, alhamdulillah. So can you repeat the reason why this happens? I absolutely can, yeah. So basically, once we said that the potassium channels are going to open somewhere between 3 to 10. And once your potassium channels open, they're going to start leaving the cell. And this will decrease the positive charge of the cell, okay? But something about the cardiac myocytes is that you don't want the action potential to wait, why is this not writing? Okay, something about cardiac myocytes is that you don't want the action potential to occur like this. You don't want it to occur very quickly. You want them to have a little plateau phase over here because you want to make sure that the contractions last long and they're strong enough. So if you have like tiny contractions right after each other, you won't be able to pump enough blood outside of the heart, And even more, you won't be able to fill the blood up with enough, you won't be able to fill the heart up with enough blood, okay? So again, the reason why you can't have these kind of action potentials in a cardiac myocyte is because you want enough time for a full contraction to occur, a full strong contraction to occur, which is why we have this plateau phase. And how does this plateau phase happen? Once again, we said between 10 to 30 millivolts, we have potassium channels opening. Potassium ions begin leaving the cell. And once potassium ions leave the cell, you're going to reach like 5 millivolts, which is when the L-type calcium channels will open. If the L-type calcium channels open, they're going to enter the cell, which is going to cause like an equal amount of potassium leaving and calcium entering. Did that answer your question? Okay, great. So over here, you guys can see like a statement that says myocytes cannot be tetanized. What does that mean? How do I know during which phases will the gates be closed or inactivated? Okay, I think this is kind of something you have to like practice and memorize. So it's kind of like a pathway, like one thing will lead to the other. So I'm just going to recap it very quickly for you, and you tell me if there's any specific point that's confusing you. We said phase 4, you're at your normal conditions, nothing open just yet. But then once you get a signal from your SA node, this is going to cause, okay, once you get a signal from your SA node, you're going to get depolarization until -55. The negative 55 millivolts is a signal for your voltage-gated sodium channels to open. So once you reach this level of electrophysiology, you're going to get a signal to open the voltage-gated sodium channels, okay? And that's going to cause an upstroke all the way until you reach the 30 to 10 millivolt region. Once you reach this region, you're going to get a signal to open the potassium channel ions, potassium ion channels, okay? And once this opens, then it's going to decrease back down to 5 millivolts, and that's going to cause the calcium channels to open. Do you kind of get it now? They're kind of signaling each other to open. So you open this channel, and then the resting membrane will change, and then that's going to cause another channel to open. So that's how it works. Is that clear? Okay. So moving on, we stopped at, is it like a regular action potential? It is kind of like a regular action potential, except that you do not have this plateau phase in a normal action potential. So yes, if you understand how a regular action potential works, it's basically the same mechanics. Okay, perfect. Let's move on. So I stopped at this part. Myocytes cannot be tetanized. I don't know if you guys are familiar with the term tetany or tetanus, but basically tetanus just means like tiny contractions that occur back to back that can't be stopped. So they're kind of like spasms, and they can happen because of like a bacterial infection or just like some form of diseases. The point is, because this plateau phase exists, myocytes can never undergo tetany because you have this plateau phase that prevents you from having another contraction, and it prevents the contractions from happening very quickly. Okay, that's all there is to it. So moving on, phase 3. So at some point, the calcium channels on their own, like after a specific period of time, the calcium channels will close. So now the calcium channels close. Here, there's no like specific signal, just on their own after a certain period of time, they will inactivate and they will close up. And since you closed up the calcium channels, now you just have a bunch of potassium leaving the cell. And when you have a bunch of potassium leaving the cell, then you're going to decrease the positive charge all the way until you reach the end part of phase 3. At the end of phase 3, you're going to open another potassium channel called the IK1, okay? So the first one that we opened was the transient outward. Now we opened something called the IK1 in late phase 3. In the end, they both do the same thing. They both kick potassium outside of the cell until you reach the normal resting membrane potential, which is -90 millivolts. So this was like a huge mouthful. Do you guys need me to repeat any parts before we move on? Yes, I can. So basically, we just finished phase 2. We just finished the plateau. We just finished the calcium ions entering the cell. On their own, the calcium channels on their own, they're going to close back up after a specific period of time. And once they close up, you're going to have a lot more potassium leaving the cell. Like now you don't have potassium is leaving, but calcium is entering, so they're balanced. There's no more balance. Your potassium stopped entering the cell, so you just have a bunch of your calcium stopped entering the cell, so you just have a bunch of potassium leaving the cell. And this is going to cause your cell to become more negative, until you reach the end of phase 3, where you have another potassium channel opening, which is called the IK1. The IK1 is going to cause even more potassium to leave the cell. So now your cell is becoming even more negative until you reach the resting membrane potential. Is that clear? What was the other thing? Oh, the other thing that was like IK1, I was just telling you in phase one, you differentiate between the channels. Phase one, you're opening the transient outward potassium channel. But in phase three, in late phase three, you're opening the IK1. Both of them are potassium channels, but they're different ones. Can a second potential action potential happen in phase three? Yes, it can, but in a very specific way. We'll discuss it in the next slide. Other than that, do we have any questions? Okay, so let's move on to, remember something. I remember the number negative 70 MV from So What Happens Here. I think, okay, I did not have access to recordings or transcriptions for this lecture. So unless he specified that at negative 70 millivolts, the IK1 will open, I can't really confirm or deny that. So unfortunately, you guys are going to have to check whether at negative 70 millivolts specifically, the IK1 will open up. Okay, so, okay, thank you. Let's move on to refractory periods. And I think that's what one of your colleagues was asking about. So a refractory period means that in this specific period of time, you cannot elicit another action potential. You have two different types of refractory periods. You have your absolute refractory period and your relative refractory period. So remember how we were talking about the sodium channels in phase zero? What's going to happen is, as I said, they're going to inactivate almost immediately, like within milliseconds, they inactivate. Once they are inactivated, once the sodium channels are inactivated, you cannot have another action potential. Again, remember, if I told you, if they ask you what causes depolarization, what causes the action potential, you do have to say sodium. So if the sodium channels are inactivated, that means you absolutely cannot have another action potential. So, and the sodium channels will remain inactivated until phase two is over. So during this period of time, they have it written over here. During this period of time, this is called the absolute refractory period. And you cannot have another action potential at all, ever, no matter what, okay? But then you enter phase three. And in phase three, what's going to happen is that your sodium channels move from inactivated to closed. So now they're not inactivated, they're activated, but they're still closed, so no sodium is entering. In this case, you can have an action potential, but you're going to need a much larger stimulus. So it's not going to be your normal stimulus that usually causes the depolarization. You need a very strong stimulus to cause them to open up at this stage. And this occurs in phase three all the way until you begin phase four. Once you guys reach phase four, you're back to your normal resting membrane potential. You just need a normal stimulus. So to answer your question, can a second action potential happen in phase three? Yes, but it needs a stronger than normal stimulus. And I think this part's pretty simple. It's the exact same concept from your action potential lecture. Let's move on. In this slide, all he needs you to know is that the refractory period of the cardiac muscle is for around like 200 milliseconds. And as we said, in the absolute refractory period, you can never have an action potential. In the relative refractory period, you can, but you need a larger than normal stimulus. And one more thing is that once you get a signal to a cardiac tissue, once you get a signal to a cardiac tissue, you do not want the cells to contract individually. You need them to all contract in the same time at the same way, okay? And this is what we call a functional syncytium, when all the cells contract as one singular tissue, as if they're one singular cell. Because obviously, if you have every cell contracting at a different time, you will not get a productive heart contraction where the blood is being pumped out. So for this part, just remember what a functional syncytium means. And for the graphs, as I remember, there's nothing special to remember. Just remember that this is what an action potential looks like for a skeletal muscle, and this is what an action potential looks like for a cardiac muscle. Don't confuse them. Do you have any questions before I move on? Okay, let's move on. Who can answer this question for me? Yes, perfect. It's B, potassium. What determines the resting membrane potential is B. Now, second question. Yep, it is B, sodium. Anything that's like what causes the upstroke, the depolarization, all of that, it's sodium. Okay. Now we want to talk about excitation contraction coupling. What does the phrase excitation contraction coupling even mean? Excitation, we're talking about the electric activity. So we're basically talking about the action potential. Contraction, we're talking about the literal physical contraction. So basically what this phrase means is how do you link the action potential with the actual physical contraction of the heart? So What you need to know from this slide is that cardiac cells have a special structure called T-tubules. This is not found in smooth muscle cells, okay? Cardiac cells have a special structure called T-tubule. And what does this T-tubule do? It ensures that the action potential spreads across the cell very quickly, okay? So one more time, T-tubules transmit the electric signal very quickly. So let's take a look. How do we turn action potentials into contractions? What's going to happen is that you will have the action potential across the membrane. And then, as we said, we're at phase 4, and then you go to phase 0, and then you go to phase 1, and then you reach phase 2. And phase 2, we said what opens at phase 2? We said the L-type calcium channels open at phase 2, and they begin allowing calcium inside the cell. Now, what exactly happens with this calcium? It enters the cell. What does it do, right? So before we get into that, I just want you to note that the L-type calcium channel has another name called the dihydropyridine receptor. So DHP and L-type calcium channel and voltage-gated calcium channels, all these three names are talking about the same channel. So just keep that in mind because I know, they can get confusing. These three, same exact channel. It opens up in phase 2, as we said, and then the calcium will enter. What will the calcium do once it's inside the cardiac cell? It's going to go to the sarcoplasmic reticulum. And at the sarcoplasmic reticulum, you have a receptor called the RYR, the ryanodine channel, okay? And it's going to bind to the ryanodine channel. Once it binds to the ryanodine channel, this calcium is going to open up this channel, and now you have even more calcium being released from the sarcoplasmic reticulum, okay? So basically, the calcium is forcing more calcium release. Calcium will enter the cell from the L-type calcium channel, or the DHP receptor, and it's going to enter the cell. Once the calcium enters the cell, it's going to bind to RYR, found where? Found on the sarcoplasmic reticulum. Once it binds to the RYR, it's going to open the channel up, and it's going to cause more calcium to leave into the cytosol. And this is going to cause something called a calcium spark, just a bunch of calcium building up in the cell. And then that's going to cause a calcium signal. And then this calcium signal is going to go bind to troponin, which is found in our sarcomere, okay? Is this clear to everyone? Okay, I'm assuming it is. So once it binds to troponin, we're going to enter something called the cross-bridge cycle. And you're going to have to wait on the cross-bridge cycle just for a moment because we have to explain how you relax first. Like, how do you get the calcium out first? So just keep this in mind, okay? Once you get calcium in the cell and it's released from the sarcoplasmic reticulum, it will bind to troponin, do the cross-bridge cycle, and then now we're going to discuss relaxation, okay? So you're done with the cross-bridge cycling, you're done with contracting, and you want your heart muscle to relax. How does it exactly do that? For it to relax, you need to get the calcium back to its normal places, either back into the sarcoplasmic reticulum or back outside the cell over here. So you could do that in two different ways. If you want to get into the sarcoplasmic reticulum, you're going to need an enzyme called SERCA. Okay, so the sarcoplasmic reticulum has an enzyme or has a channel on its membrane called the SERCA. But the thing about SERCA is that it has a regulatory protein called phospholamban. Okay, just this one over here. It has a regulatory protein on it called phospholamban. And normally, under normal conditions, the phospholamban is inhibiting SERCA. So let's say you have calcium here, and you have phospholamban attached to SERCA, and you want to put this calcium back to the sarcoplasmic reticulum. Under normal conditions, this phospholamban is inhibiting SERCA, and you cannot get the calcium back inside the sarcoplasmic reticulum. So what do you need to do to get this phospholamban to stop inhibiting SERCA? You're going to need a phosphate molecule. Okay, so here we have P, whatever, the phosphate. You don't need to know how it got phosphorylated. You just need to know that it needs to be phosphorylated. Okay, so the phospholamban will get phosphorylated, and once it's phosphorylated, you're going to remove this inhibition effect by the phospholamban. And what's going to happen is that the SERCA can actually push the calcium back into the sarcoplasmic reticulum. Is that clear? Can I get some form of thumbs up, or do I need to repeat? Okay, alhamdulillah. Now, we successfully did our first mission, which is to put the calcium back into the sarcoplasmic reticulum. Now we need to get the rest of the calcium back outside the cell because we didn't get it only from the sarcoplasmic reticulum, we also got it from outside of the cell. This is why you have another enzyme, which is called the NCX, which stands for sodium calcium exchanger. Okay, so sodium calcium exchanger. And from the name, what it's going to do is that it's going to take 3 sodium into the cell while pushing out 1 calcium. and that's how it kind of kicks the calcium back outside the cell. But there's an issue here. The issue is that under normal conditions, you have low levels of sodium inside the cell, and here is like outside. Okay, so you have high levels of sodium outside the cell. But because of this exchanger, because you're trying to kick the calcium out, the amount of sodium inside the cell is going to increase. And that's not good. You do not want the amount of, like under normal conditions, since you're trying to relax and you're trying to go back to your basal state, you don't want your sodium to increase because that's going to ruin a lot of gradients and that's going to cause a huge mess inside the cell. So what we need to do is we need to decrease this amount of sodium, like ASAP. How do we do that? This is where the sodium potassium pump comes in. Okay, and this is a very important enzyme that you're going to see in like a million different ways. So what this sodium potassium pump is going to do is that it's going to kick those 3 sodiums back out. So, you're not having this increase of sodium inside the cell, and it's going to bring back 2 potassium inside the cell. You guys might be asking, but like, why is it bringing potassium inside? Like, what's going on? It's fine if you have an increase, like if you need to have a high amount of potassium inside the cell. That's the normal condition. You usually have a higher amount of potassium inside the cell. So is that clear to everyone? Do you understand how the NCX and the sodium potassium pump works? Okay, amazing. So the conclusion from here is I want you to remember that, number 1, if phospholamban is not phosphorylated, then SERCA will not work. So if phospholamban is not phosphorylated, SERCA will not be able to work. So you need to phosphorylate phospholamban. And for the NCX exchanger, what I need you to remember is that if the sodium potassium pump is not working, then this is going to affect the NCX exchanger, and it's also not going to work. So just keep that in mind because we're going to discuss them again in the future. This is just a very quick summary slide. To relax the muscle after contraction, calcium must be kicked out. You can either bring it back into the sarcoplasmic reticulum through SERCA, or you can bring it back outside the cell through the sodium NCX exchanger. And then one final detail is that inside your sarcoplasmic reticulum, you have a protein called calsequestrin. What does this protein do? It holds on to the calcium. So remember how we were like, oh, the sarcoplasmic reticulum stores a lot of calcium. How does it store a lot of calcium? Through this protein called calsequestrin. Very easy name. Cal is for calcium, sequestrin is for storage, so calcium storage protein. Quick checkpoint. Who can answer this question? Yep, B. That's it. One more question. C, exactly. Perfect, you guys got it. It is C. And for those who are confused and you're like, what? Calcium entry happens by the L-type calcium channels. As we said, the L-type calcium channels are the DHP channels. Literally the same channel, just 2 different names. Okay, now, based on everything we learned, we can kind of discuss some factors that affect the myocardial contraction. So in other words, what are the factors that are affecting the ability of our heart to contract? So one thing you need to keep in mind is that when you have a myocardial infarction, which means the death of your heart tissue, myocardial means, heart, infarction means death, okay? So when you have an MI, 2 things are going to happen. Number 1, you're decreasing the amount of oxygen in this tissue, which means you're going to decrease the amount of ATP produced because of oxidative phosphorylation, okay? So in a myocardial infarction, you have less oxygen, which leads to less ATP. And at the same time, you have an increase in hydrogen ions. Both of these, both the decreased ATP and both the increase in hydrogen will affect the DHP. And if you affect the DHP, then that means calcium cannot enter the cell anymore. And if calcium cannot enter the cell anymore, then no contraction. So very simply, any muscle any part of the heart that undergoes an MI can no longer contract. It's damaged. Is this clear? It's very simple. I think it is clear, honestly. Okay. So moving on, what about the sodium calcium exchanger? As I said, the sodium calcium exchanger, the NCX, directly depends on the sodium potassium pump. And we have a drug called digitalis, okay, digoxin. And what this drug is going to do is that it's going to inhibit the sodium potassium pump. So if this is inhibited, okay, then this is going to inhibit the NCX. If the sodium potassium pump is inhibited by digitalis, then that is going to inhibit the sodium potassium pump. Now, this does not mean that you won't have contractions. quite the opposite, actually. So you guys need to remember what NCX does. NCX, as we said, brings 1 calcium outside of the cell and 3 sodiums inside the cell. Why are we kicking calcium outside the cell? Because if you remember, we said we need to get this calcium out because we want to relax. Okay, so if you want to decrease the calcium because you want to relax. So if this NCX is not working, what's going to happen is that this calcium is going to stay inside the cell. And that means you're increasing your calcium concentration. And if you're increasing your calcium concentration, then you will not relax. Instead, you will contract. So in other words, what does digitalis do? What does this drug do? It inhibits the sodium-potassium pump, which will affect the NCX exchanger. And because of the buildup of calcium, you're going to have stronger contractions. Is that clear? Again, absolutely. Okay, one more time. We have a drug called digitalis or digoxin, same drug. And what this drug is going to do is that, let's put this here, digoxin. And digoxin is going to inhibit the Na potassium pump, the sodium potassium pump. And we said the sodium potassium pump will directly affect the NCX exchanger. Let's go back up to the slide we were explaining, right? We said this one, if this one does not work, then this one will also not work. And what happens if this one does not work? If the sodium calcium exchanger does not work, then the calcium will not leave the cell. It will just build up inside the cell and it will increase the amount of calcium in the cell. And if you have a lot of calcium inside the cell, you will not be able to relax. You will just have stronger contractions. Is that clear? Okay, perfect. One last point, which is the role of phospholamban, which we already discussed. As I said, phospholamban, this is phospholamban. Let's represent it by PL, okay? PL, that's phospholamban. Phospholamban usually inhibits SERCA. This is SERCA. And if SERCA is inhibited, then you cannot put the calcium back inside the sarcoplasmic reticulum. You need to phosphorylate phospholamban to allow the calcium to go back in easily. There's nothing more to it on this slide. Do you have any questions? Okay, I think we're good. Who can answer this question for me? Yes, it is C. Phospholamban is not phosphorylated, meaning that the calcium will not be able to return to SERCA, meaning that the calcium levels will just increase inside the cell. So you guys got it, it is C. Now we are moving on to cross-bridge cycling. Inshallah, after we're done with this concept, I will be giving you guys a quick break. Okay? So to begin with cross-bridge cycling, I will go over the thick filaments and the thin filaments that we discussed earlier. We said thick filaments are your myosin. So thick filament, your myosin, okay? And then over here, you have your myosin chain. And then you have your 2 myosin heads. So wait, let me just write heads. And what did I say that the heads have? They have ATPase activity. Very important to remember, okay? And then over here, you have your actin. Well, not that molecule, your thin filament. Your entire thin filament is your actin. And then you have tropomyosin, and you have troponin, T, C, and I. Okay, I know these are a lot of names, and some of you guys might not fully understand them. If you can just understand that these are the components that you're dealing with, that's enough. So once again, thick filament, myosin. What does myosin have? It has 2 heads. And what do the heads have? They have ATPase. Thin filament, what's our thin filament? Actin. And what does the thin filaments have? They have alpha tropomyosin. They have 3 different types of troponin. Okay, now let's talk about the state that you're in when you're resting. How do these filaments act when you're resting? You need to focus on this half of the slide right now. When you're in your resting state, the myosin head will be at 90 degrees, okay? So this is what they mean. I don't know if you guys studied this lecture before attending the PAL, but if you did, you might have noticed the phrase 90 degrees and you might have been like, what does he mean by 90 degrees? This is what he means. This is the 90-degree angle. And furthermore, the myosin head has your ADP and phosphate attached to it. When your myosin head is at 90 degrees and when it has ADP and phosphate attached to it, you know that your myosin head is in its resting state. Okay, so now we know that the myosin head is in its resting state. Furthermore, we can see that there's a structure called tropomyosin that is preventing your myosin head from directly attaching to the actin molecule. Okay, so we are in this is where you guys start your cross-bridge cycle. Your myosin head has ADP and phosphate attached to it. It's at its low energy, 90 degrees state. and the tropomyosin is inhibiting it from directly binding to the actin. So what happens? What changes? Remember how all the way in the slide, wait, let me go back. Right, in this slide, I said you have a calcium signal. Okay, yeah. I said you have a calcium signal over here that's going to go bind to something called troponin. This is where we're starting the cross-bridge cycle from, okay? So, wait, let me just go back to the original slide. Right, so because of everything that we talked about, because of the release from the sarcoplasm, you have a lot of calcium in your cytosol, and this calcium is going to go and bind to troponin C specifically, okay? So if you don't get confused, it's very easy. Troponin C, the C is for calcium. So it's going to go bind to troponin C, and troponin C plus something called troponin I, both of them are going to work together to pull this tropomyosin back. So remember how we had this tropomyosin getting in the way over here, like right here, getting in the way? The troponin C and troponin I are both going to pull the tropomyosin back. So now your myosin head can actually directly bind to the actin. That's all that's happening here. That's all that this slide is saying. Okay, so let's just take a look at it one more time. In stage 1, you're at rest, and the myosin head has ADP and phosphate bound to it. Furthermore, it's at 90 degrees. Okay, and this is when stage 1, you're basically at rest. And then we had that, calcium-induced calcium release. We had the calcium spark, whatever you want to call it. You have calcium that's going to go bind to troponin T and C actually, T and C plus T and I, and these are going to move tropomyosin. Once you move tropomyosin, you can actually directly bind to the actin, and that is exactly what happens in stage 2. All simple. So what happens after that? So in stage 2, we said that your myosin head will directly bind to the actin, but it still has the ADP and the phosphate attached to it. So once again, we're still at phase 2. Don't jump to stage 3 just yet. We said that the myosin head is at 90 degrees and it has ADP plus phosphate attached to it. The only thing that changed is that now it's directly bound to the actin. Okay, how do we get this thing moving? How do we get the power stroke? What's going to happen is that your phosphate and your ADP will be released. So remember how we had ADP and phosphate attached to it in stage 2? In stage 2, in stage 3, the phosphate and the ADP will be released and they will detach from the myosin head. And this is going to cause the conformational change. So once again, stage 3, release of ADP and phosphate changes from 90 degrees to 45 degrees. And you can even visualize it. Like, look at how the angle changes. It went from 90 to 45, okay? And this is what causes the power stroke. So if they ask you what causes the power stroke, the answer should be the release of ADP and phosphate. What causes the conformational change? The release of ADP and phosphate. Very easy. Moving on to stage 4. What happens in stage 4? What they're saying in stage 4 is that if you do not have ATP, if ATP does not come in and attach to the myosin head, you will be stuck in this state. And when you get stuck in the state, it's called a rigor state. That's all that's happening in phase 4. Nothing new. Okay, so the actin will remain in its position, the myosin will remain in its position, and now what you're in is called a rigor state until stage 5 happens, where ATP comes in. Once ATP comes in and binds to the myosin head, you'll have detachment. Okay, and remember how I said myosin head has remember how I said the myosin head has ATPase activity? I don't think I wrote it here. Myosin head has ATPase activity. What does ATPase mean? ATPase, just give the screen some time to catch up. Okay, what I was saying, myosin heads have ATPase activity, and I kept saying this multiple times throughout the lecture. Why? Because this is where it comes in right now. ATPase is just an enzyme that hydrolyzes ATP into ADP and phosphate. So after the ATP attaches to the myosin head and it causes it to detach from the actin, the ATPase will actually hydrolyze it back into ADP and phosphate. And this is how you guys land right back to your original state, okay? So here's what I was saying through the diagram. ATP attaches to the myosin head, causes detachment. And then because of the ATPase activity, you'll have hydrolysis, and now you're back at stage 1. I think I do need to repeat this part, but just can I get a confirmation if everyone is with me on board right now? Okay. would you guys like me to repeat any specific part, or are we good enough to move on? Okay, perfect. I'm glad that you guys are following. So just one last time to recap what the role of ATP is in here, because I think a lot of MCQs come from this concept. In your resting state, your ADP and your phosphate are bound to your myosin head. Okay, but then calcium comes in and it causes the myosin to bind to actin. Once the myosin binds to actin, the ADP and the phosphate will release, and this is what causes the conformational change, and this is what causes the power stroke. So if they ask you what causes the power stroke, again, it's ADP and phosphate release. And then you enter the rigor state until the ATP comes in and forces your myosin to detach. And then what happens is that your myosin has ATPase inside of it, which will hydrolyze that ATP back to ADP and phosphate. Once it's hydrolyzed, you're basically back at stage 1 and you're ready to repeat the cycle again and again. Do you have any questions before we continue? Clear, perfect. Please answer this question. Yep, absolutely, it is B. Okay, so for those, let's just walk ourselves through it real quick. They're just telling you to organize the order in which these actions happen. First of all, you're going to have your action potential. After the action potential happens, you will have the depolarization of the T tubules, which will cause the calcium to enter the cytoplasm. Once the calcium enters the cytoplasm, you will have cross-bridging. Once you have cross-bridging, you want to relax, so you'll have calcium removal. Very easy, very straightforward. And, oh, one more question. Yep, absolutely, it is B. Another question? Yes, it is C. You guys are doing great. We're going to begin with the next lecture, but you guys do deserve a quick break. So how much do you guys need? 5 or 10 minutes? Because I know you're probably exhausted. 5 minutes, perfect. I'll meet you at 4:17 then. I'll meet you guys at 4:17. Go ahead. All right, perfect. So moving on to smooth muscle contractions. Once again, we're just going to begin with a brief, description of what smooth muscle cells are. Once again, they are contractile cells, but their contractile apparatus is not in sarcomeres. So basically, they do have the same structure. They do have thin and thick filaments. They're just not arranged in sarcomeres, which is, they're not arranged in like the A band, I band, that kind of thing. Second of all, they are fusiform. Remember how we said the cardiac cells are branched? They're fusiform. What does fusiform mean? It means this shape, yani, this basically wavy shape. And you could see it basically on the histological photo, yani. That's what fusiform means. They're non-striated, so they do not have those dark and light bands. As I said, they have a single central nucleus. They undergo involuntary contraction by neuroendocrine chemicals. So remember how we said the heart has pacemaker cells? Smooth muscle cells do not have pacemaker cells. Instead, neuroendocrine chemicals by the autonomic nervous system is going to cause them to contract. And finally, they can be found in blood vessels, GI organs, urogenital organs, in your eye. They're found everywhere, basically. Now, in your PowerPoint, he put 2 slides with a bunch of information. If you guys were like cramming for the exam, I highlighted what I think is the most important information from the slides. So let's just go over them really quickly. Number 1, smooth muscle actin contains tropomyosin, but does not have troponin. So it does not have troponin C or troponin I, but it does have tropomyosin. Second of all, the cytoskeleton of the smooth muscle contains intermediate filaments and protein-dense bodies. I think you do need to remember this, the protein-dense bodies being found in smooth muscle. That's an important piece of information. And I think it's repeated in histology too. Cytoskeletal fibers will link the dense bodies to the cell membrane. Another important point to remember about dense bodies. And then finally, smooth muscles do not have T-tubules. Instead, they have something called caveolae, which are like invaginations, okay? They're also supposed to help with the cell signaling. But just remember, Yani, instead of T-tubules, we have caveolae, and they kind of do the same function. So smooth muscles can be classified by location, by contraction pattern, and by the way they communicate with their neighboring cells. Location, we're not going to go into that. We just briefly discussed where, smooth muscles can be found. But for contraction pattern, we have 2 types. You have your phasic contraction and you have your tonic contraction. Phasic, from the name, you're undergoing a contracted phase and then you're undergoing a relaxed phase. Why is this useful to us? Why, subhanAllah, why do we have this in our body? Because we have, for example, we need this in peristalsis. You want to push the food down, so you want to contract and then relax, contract and then relax. This also happens in the intestine to allow for the mixing and processing of your food. And then what about tonic contractions? Why do we have tonic contractions in our body? Tonic contractions are usually found in sphincters. So you have something above your stomach called the pyloric sphincter. And the pyloric sphincter sits between your esophagus and your stomach. And it's always closed unless you're obviously trying to pass a food down to the stomach. Why is it always closed? Because if it was open, or if it was weakly closed, then you're going to have the acid go back up through your esophagus. And that's kind of what happens with the disease GERD, if you guys ever heard of it. Okay? So once again, smooth muscles can either contract phasically, so like seen in the esophagus and in the intestine, or they can contract tonically, which means they're always contracted, such as with your pyloric sphincter. And then he gave you 4 different graphs. I honestly don't know how he would ask you about this. Like, I don't think we've ever gotten a question on these graphs, but we can just go over them. They're just examples. So here you have a phasic smooth muscle that is usually relaxed, your esophagus. Your esophagus is not constantly undergoing peristalsis, right? It's usually relaxed until you get food, and then that's when the wave starts to happen. And then you have a phasic smooth muscle that cycles between contraction. So this one is like in your small intestine, you're constantly going peristalsis. It's constantly contracting and relaxing. But then you also have, he gave you a graph that's for a tonic smooth muscle that is usually contracted and then you suddenly relax for a specific purpose. So once again, your pyloric sphincter between your esophagus and your stomach. You eat something, you want to push the food down your esophagus to your stomach. So that's going to cause the pyloric sphincter to relax, and then it immediately goes back to contracting. One last example is the vascular smooth muscle. So your blood vessels, they're not always relaxed because otherwise you would be hypotensive and you would faint. And they're obviously not always like at maximum contraction, because otherwise you would be hypertensive and you would, I don't know, it's not good for you. Okay, so instead you're at this like medial contracted tonic phase, and then once, let's say you stood up or you did a specific action that causes your blood vessels to relax a bit for whatever reason, and that's when you're going to have the fluctuations. So instead of being like a straight line, your blood vessels are kind of going to like stay contracted at this level, but then they're going to relax for a specific reason. And I don't know, so on. I don't think these graphs are important, but just for the sake of completion. Moving on, communication styles. Your cells are either going to communicate as a single unit or they're going to act as multi-units. What does that mean? So single unit can be typically seen in the walls of the intestinal tract. And how exactly do we get the intestinal tract to act as a single unit? So the way your small intestine is arranged is that you have your autonomic neuron on the inside, and then you're going to have a bunch of layers of smooth muscle cells next to it. And the autonomic neurons are going to release those neuroendocrine chemicals that we spoke about earlier, and they're going to come and bind to the first layer of smooth muscles, this layer. So, and this is going to cause them to depolarize and contract. But you might ask yourself, type, if the neurons are only touching this layer of smooth muscle cells, how is it reaching all the other layers? This is through the gap junction. So it kind of acts the same way as the cardiac cells. You remember how cardiac cells have the intercalated discs in between each other? They don't have intercalated discs. Don't get those two confused. Instead, they have gap junctions, which act similarly. They allow the signals to spread throughout the tissue, and therefore all of your smooth muscle cells will contract at the same time, causing for peristalsis to happen. And that's what single unit means. So what about multi-unit? Multi-unit can be typically found in the iris or in the ciliary muscles of the lens. And the way this acts is that you have your autonomic neurons. They're going to be spread out between every single cell. This way, every single cell will contract with a specific force and in a specific direction. And the way I want you to think of it is that your eye is a very precise organ. You need to move in a very precise way. You need to allow a very precise amount of light inside, which is why you're going to need this multi-unit communication, because you want to be very specific with your movements. So, you individually control every single muscle. And yeah, that's pretty much it. Before we move on, who can answer this question? It's a simple recall question. So even if you don't get, it's C. And even if you don't get it now, if you just go back to that one slide, you'll see it. But it is C, so good job. Now let's move on to the contractions of smooth muscle. In general, whenever they ever ask you about contractions, like what ion causes contractions, it's calcium. Okay, so you need calcium to make your smooth muscles contract the same way you need calcium to make your cardiac muscles contract. And where is this calcium coming from? Once again, this calcium is coming from the extracellular compartment and the sarcoplasmic reticulum. But this is going to happen in a slightly different way than the cardiac cell. So at the level of the sarcoplasmic reticulum, first of all, remember, we do not have this T-tubule stuff, right? So at the level of the sarcoplasmic reticulum, you're going to have your ryanodine receptor, the RYR receptor, which is going to open in response to calcium and cause the calcium-induced calcium release, the same way as a cardiac cell. So this one, it's found in both cardiac cells and smooth muscle cells, okay? But then your sarcoplasmic reticulum in your smooth muscle cell is also going to use the IP3 receptor. Now, the IP3 receptor I think it should have been discussed in a different lecture called the cellular communication lecture. So you guys are supposed to be familiar with the whole G protein, phospholipase, BIP2. Can someone confirm whether you took this in another lecture or not? Okay, so I got a nope. In any case, the doctor did not go into it in detail, so I don't think you need to know the exact pathway. What I do need you to know is that in your smooth muscles, you mainly depend on the IP3 receptor to release calcium from your smooth muscle, okay? So you're supposed to have an entirely different lecture on this. He did not even go into details in this lecture, so apologies, but we don't have enough time to cover this entire pathway right now. What I do want you to know, smooth muscle cells needs both the ryanodine receptor and the IP3 receptor to release calcium from the sarcoplasmic reticulum. But it mainly depends on the IP3 receptor. So this is the primary one. This is like, let's put primary, okay? But it also uses the ryanodine receptor. In cardiac cells, you do not have the IP3 receptor. So keep this as a distinction, because he might get you an MCQ, which one is found in the cardiac cells, which one is found in the smooth muscle cell, okay? So now we've achieved the same thing. Okay, we got through those two receptors, we got calcium outside of the sarcoplasmic reticulum. Now we have calcium in the cytosol. What is going to happen from there? This calcium is going to bind to a protein called the calmodulin. Once again, this calmodulin protein is only in the smooth muscle. So this is a completely different pathway only in smooth muscle cells. This does not happen in cardiac muscles, okay? Calcium will come and bind to calmodulin. What is this calcium calmodulin complex going to do? This calcium calmodulin complex is going to go and activate an enzyme called the myosin light chain kinase. Okay, so once again, what will this myosin light chain kinase do? I don't know if you guys are familiar with this, but basically, kinase is a very specific type of enzyme that phosphorylates. Okay, it will always phosphorylate. Whether this phosphorylation has like a inhibitory action or stimulatory action, that depends on the molecule. But point is, a kinase will always phosphorylate something. So what will this myosin light chain kinase phosphorylate? From the name, it will phosphorylate the myosin light chains. And once it phosphorylates them, it's going to activate the myosin light chain. Okay, and this will cause it to activate myosin ATPase activity. I know we just said a bunch of things all at once, So let's just let's just review what I said real quick. You have from your extracellular matrix from outside of the cell and from your sarcoplasmic reticulum, you're going to get a bunch of calcium into your cytosol. And once you get a bunch of calcium into your cytosol, you're going to bind to this protein called calmodulin. Once you bind calcium and calmodulin, you're going to go and activate your myosin light chain kinase, okay? Now it's activated. And what did we say this myosin light chain kinase will do? It will go and phosphorylate the myosin light chain. Okay, so let's go back to this diagram now. Once again, I put the diagram of the thick filaments and the thin filaments for you. This is your thick filaments. This is your myosin. And then this is your actin. Well, not this specifically, this whole thing, okay? This is your actin. And as I said, your myosin light chain kinase will go and phosphorylate the myosin heads with the myosin light chain. And once you phosphorylate them, you're basically activating them, which is very important for you guys to remember. So what will activate the myosin light chain? What will activate the myosin ATPase? The MLCK phosphorylation. I'm going to say that one more time. What activates the myosin head light chain and the ATPase? The MLCK phosphorylation. Okay? So up until this point, do you guys have any questions? Should I repeat this process one more time? Yes, please. Absolutely. So once again, when it comes to smooth muscles, you do have a different pathway to reaching the cross-bridge cycling, because we're doing all of this because we want to reach cross-bridge cycling. So the smooth muscle way is a bit different than the cardiac muscle way. First of all, we're going to get calcium. Obviously, for a contraction to happen, for the cross-bridge cycling to happen, we want calcium. And where are we going to get this calcium from? The calcium is going to come from two places, the extracellular matrix and the sarcoplasmic reticulum. And we briefly discussed how does the sarcoplasmic reticulum give us the calcium in smooth muscle cells in two ways. Number one is the RYR receptor. Number two is the IP3. And if anything, it's mainly the IP3, but it's also the RYR. Okay, perfect. Now we have a bunch of calcium in the cytosol. Once this calcium is in the cytosol, it's going to go and bind to a protein called calmodulin. Calmodulin is only found in smooth muscle cells. Okay, now they're going to go and form a complex, the calcium calmodulin complex. Once they form this complex, they're going to go and activate something called myosin light chain kinase. And as I told you, kinase is just an enzyme that will phosphorylate something. So calcium calmodulin complex will go and activate the myosin light chain kinase, which will go and phosphorylate the inactive myosin. And now you have an active myosin. Very simple, okay? And this, I just showed you a diagram of what the myosin looks like. So let me just clear this part really quick to go over it again. I was just telling you, this is the thick filament. This is the myosin. This is the thin filament. This is the actin. The MLCK will come and phosphorylate this. It's going to go and phosphorylate the light chain, which is going to go and activate the ATPase. And as I said in the previous lecture, where do we find the ATPase? In the myosin head. So basically, once you phosphorylate the structure, you're activating the myosin. Is that clear for everyone? I can repeat if anybody wants me to repeat. Okay, perfect. I think we are clear, unless someone asks me to repeat. Okay, just to check, can you guys quickly solve this question for me? Yes, perfect. Amazing, you guys got it. It is A, perfect. So it's phosphorylates myosin, therefore activating it. And once you're activating myosin, khalas, now you can begin cross-bridge cycling, which is exactly what we're going to talk about. And this is very simple. It's very similar to what we did in the cardiac cell lecture, but it's just the beginning that's different. So I'm going to go over the beginning again. At this stage, and here's one. Okay, so before we enter step one, we said we have the myosin light chain kinase, which is going to go and phosphorylate the myosin. Where is the myosin? Over here. Okay, now that it's phosphorylated, it's basically in its high energy state, but at the same time, it still has the ADP and the phosphate that we spoke about in the cardiac cell lecture. You know how we said in the cardiac cell lecture, you're supposed to have ADP and phosphate attached to the myosin? That part does not change. The only thing that's different here is that how did we activate the myosin head? How did we get it in its high energy state? We used the myosin light chain kinase to phosphorylate the myosin head, and now it's active. And now the myosin can bind to the actin because it's in its high energy state. Okay, and once the myosin binds to the actin, you want to cause a power stroke. How do we cause a power stroke? You need to release the ADP and the phosphate. Once you release the ADP and the phosphate, you have this conformational change, 45 degrees, same exact thing. Now you have a power stroke. And what did we say that what happens after a power stroke? It's going to stay attached in a rigor state unless ATP comes in. Okay, this part does not change. This is the exact same thing from the cardiac cell lecture. Okay, so you're in a rigor state unless ATP comes in. But then ATP comes in. ATP will bind to the myosin head, cause it to detach. But then, as we said, we have the myosin ATPase activity, which will cause it to hydrolyze this. And then it's back like over and over again. So I just briefly went over the same concept from the previous lecture. Is there any confusion in this part? I think we're good to go because it's the same exact concept. Now we have one more difference between smooth muscle cells and cardiac cells. Smooth muscle cells will have caldesmin and calponin. Because remember how we said we have no troponin? Instead, they have caldesmin and calponin. He didn't go into the details. All you need to know is that they will inhibit the myosin ATPase activity. So they're not always active. You do not need to know what activates those two proteins. You just need to know if an MCQ came. Number one, they are not found in any other cell other than the smooth muscle cell. Number two, they can inhibit myosin ATPase activity to regulate the contractions. How do they activate? When do they activate? That was not within your learning objectives. Okay. So now we're done with contractions. How do we relax in smooth muscle cells? It's the same concept from the previous lecture. You want to get the calcium back to its normal areas, either back in the sarcoplasmic reticulum or back outside the cell. So at the level of the cell membrane, where you're kicking them outside the cell, you have two enzymes this time. First one is the NCX exchanger. This one is the same one from the last lecture, okay? But then you also have another one called the plasma membrane calcium ATPase. This one, it doesn't like exchange anything. You're just kicking calcium out. You can see them in the diagram over here. So again, at the level of at the cell membrane, you have the NCX exchanger, 3 sodium in, 1 calcium out, the same exact one, and the same exact concept applies. You do need the NAK-ATPase. So don't forget that. The same thing applies here. Another thing, but except you have another enzyme here called the PMCA. PMCA will just kick out calcium. It won't bring anything in or anything special. And then at the level of the SR, you have circa. Nothing new here. Same exact concept from the previous lecture. Okay, so we kicked calcium out. We kicked it out of the cell, and we kicked it back into the sarcoplasmic reticulum. How does this bring about relaxation here? When you have less calcium in the cytosol, you will have less calcium binding to calmodulin. So once again, remember in this slide how we were talking about, oh, calcium comes in and it will directly bind to calmodulin. If you have less calcium, then there's nothing binding to calmodulin. And if nothing's binding to calmodulin, and if this does not exist, if this does not exist, then you cannot activate the MLCK. And if you cannot activate the MLCK, then none of this will happen. You will not activate the myosin ATPase, and you will not have cross-bridge cycling. Okay, so once again, what I was saying is that you will decrease the amount of calcium through the enzymes that we spoke about, which were the NCX exchanger, the PMCA, and the circa. Okay, so that's how you decrease the amount of calcium. If you decrease the amount of calcium, then nothing will bind to calmodulin. If nothing binds to calmodulin, nothing is activating MLCK. If nothing's activating MLCK, no cross-bridge cycling. Very straightforward. So back to our slide. We said, yeah, nothing's activating MLCK, so now it's going to be considered inactivated. And as we said earlier, MLCK is responsible for activating the myosin heads. Now that you've inactivated MLCK, you will not activate more myosin heads. But what about the myosin? Okay, there was something we need to phosphorylate in the SR because it inhibits. Is it also there in smooth muscles also? Yes, it is also there. Phospholamban is also there in smooth muscles. It does not change. Now, moving on, as I was saying, okay, you're welcome. As I was saying, we said MLCK is the one that activates the myosin light chain. If there's nothing to activate the myosin, oh wait, sorry, I lost my train of thought. What I was trying to say, no, we inactivated MLCK, which means we're not activating more myosin light chains. So what about the myosin light chains that we have activated already? How do we turn them off, basically? Exactly, MLCP. That's exactly what we're going to need. So, okay. MLCP. What is MLCP? MLCP is myosin light chain phosphatase. So phosphatase just stands for any enzyme that will remove a phosphate group. So again, remember, how did we even activate the myosin light chain in the first place? We used the kinase to add a phosphate group. So therefore, just logically, how am I going to inactivate it? Very simply, use a phosphatase to remove this phosphate group. So it's the exact opposite function of a kinase. So once again, MLCP will activate. How does it activate? You don't need to know. You just need to know that when you're relaxing, when you're trying to relax the smooth muscles, MLCP will activate. Once it activates, you're going to dephosphorylate the myosin light chain. Once you dephosphorylate the myosin light chain, the myosin ATPase is now inactivated, and therefore no cross-bridge cycling. Any questions? Clear. Perfect. Right. Please take a moment to answer this. Yep, it is B. Okay, perfect. So we're wrapping up the lecture. We're almost done, alhamdulillah. You have something. Okay, so right now I'm just doing a quick brief recap because we're about to enter a concept that can be a bit confusing. First of all, we said MLCK will phosphorylate the light chain, and if it phosphorylates the light chain, then you're activating the myosin ATPase, and if you're activating the myosin ATPase, you're contracting. So MLCK controls contraction. Now, what about MLCP? We said MLCP will dephosphorylate the myosin light chain, and if you're dephosphorylating the myosin light chain, then that means the myosin ATPase won't work. And then I added a statement, which was even if calcium was present. This might confuse you guys. Wait, like where is calcium coming from, right? Because we said MLCK controls contraction, but then we said MLCP will inactivate the myosin ATPase and it won't cause contraction even if calcium was present. But then we said that if calcium was present, then you have contraction. Point is, what I'm trying to say, and I understand this can be confusing, but it's a simple concept, okay? What is the concept that can be confusing? Basically, your MLCK and your MLCP exist in a ratio, okay? And this ratio is not fixed. It just depends on what the tissue is trying to do. If it's trying to relax, then you're going to have more MLCP. And if it's trying to contract, then you're going to have more MLCK. But there's a special thing that the MLCP is affecting. And this special thing is called calcium sensitivity. So let's take a moment to think about what sensitivity even means. When I tell you someone is sensitive to dust, okay, someone has asthma and they're very sensitive to dust. What does this mean? It means they can, even if they walked into a room that is clean, but then they smelt the slightest bit of dust, they will start having a full asthma attack because they're very sensitive to dust. If a normal person walks into a relatively clear room, nothing's going to happen to them because they're not sensitive to dust. This same concept applies here. When you're increasing the ratio of the MLCP, what's going to happen is that you're basically inactivating more myosin. Wait, okay. Let me just write this down for you. MLCP increases and you're inactivating more myosin. If the myosin is inactivated, then that means it doesn't matter if you have like a normal amount of calcium, right? Because the calcium is going to go and bind to calmodulin. And once it binds to calmodulin, it's going to go to the MLCK, but the MLCK cannot act on an inactivated myosin. Do you guys get this concept? Like, I'm going to say it one more time. You're increasing the MLCP more than the normal ratio. And this is going to cause a lot more myosin to inactivate. If the myosin from the very beginning is inactivated, then the kinase cannot act on it. Okay? And this is why what I'm trying to say is that the MLCP will increase the calcium sensitivity. It's because it's not going to increase the calcium sensitivity. It's going to decrease. Apologies. It's going to decrease the calcium sensitivity because you're going to need a lot more calcium to react and contract. I think I probably mixed you guys up a bit. Are you guys following? Do you need me to re-explain this concept? Yes, please. Yeah, I'll repeat it. It's okay. So once again, let's start from the very top. Okay, your MLCK and your MLCP exist in a ratio, and I'll answer your question in the end. So your MLCK and your MLCP exist in a ratio. If the MLCK is higher in the ratio, then that means you're more prone to contracting. You're going to contract a lot easily. But if your MLCP is higher in this ratio, meaning the MLCK is low and your MLCP is high, then you're going to be more prone to relaxing. But the thing about MLCP is that it will also decrease the calcium sensitivity. And then I went on to explain think about what the word sensitive means. Sensitive means that you need a little amount to do something. If you're sensitive to dust, you need a little amount of dust to get a whole reaction. So when you increase the MLCP, you're going to inactivate more myosin. So now you have more myosin that's inactivated. And if you have more myosin that's inactivated, that means even if you have like a big amount of calcium, you will not be able to have a contraction because the myosin itself is inactivated. So it cannot engage in the cross-bridge cycling. That's what I was trying to say. So therefore, that's how myosin light chain phosphatase will decrease the calcium sensitivity. Is that a bit clearer, or should I recap one more time? Okay, because I think I explained it fairly messily the first time, so my bad. Does anyone else want me to repeat this concept? Okay, alhamdulillah. Now to answer your question, if MLCP is active, why do we need calcium? MLCP, can you ask your question in a slightly different way? Do you mean if MLCP is active, then where is the calcium coming from? Okay. No, if you need to relax, then we need MLCP. So why is the calcium needed if you want to relax? The calcium isn't really needed. It's not about whether the calcium is needed or not. It's about the concept of MLCP, MLCK and MLCP existing in a ratio. So if you affect this ratio, let's say you give them a drug and it increases the amount of MLCP in this specific tissue, then that means, generally speaking, when you're trying to contract, the calcium sensitivity of this tissue is now low. It's not very sensitive to calcium. Do you understand? Does that explain it better? Could you repeat? Yeah, absolutely. Again, what I'm trying to explain through the slide is that your MLCK and your MLCP exist in a ratio. So it's not like you never have MLCP when you're contracting, and you always have MLCK when you're contracting. They exist in a different ratio. So when the tissue is trying to contract, the ratio of the MLCK will increase, and when you're trying to relax, then the MLCP will increase. So if you give the patient a drug, or if the patient has a disease or a condition that causes the MLCP to increase in this ratio, then what's going to happen is that the calcium sensitivity will decrease. So that means this tissue is now less sensitive to calcium. And that means even if you're trying to contract, because of this ratio changing, because of the fact that the MLCP is now higher than the MLCK, you will be less sensitive to calcium and you will resist this contraction. You will not be able to contract. Yes, exactly. They exist in a ratio and they're both there, but you should have it in it's either the MLCK is higher or the MLCP is higher, depending on what the tissue is trying to do. Okay. And thank you guys for being patient with me, because I know like this part was a bit messy to explain. One last time, let's recap the concept. MLCP, the whole thing that I'm trying to say in the MLCP controls calcium sensitivity. If there's an increase in MLCP, the calcium sensitivity will decrease. Why? Because you're inactivating the myosin. Therefore, you would need a lot more calcium to perform a successful contraction. And the other way around is true as well. So decrease in MLCP, calcium sensitivity will shoot up and it will be very high. And why? Because you have less inactivated myosin. Okay? Before we move on to the last two slides, do you have any questions? Clear. Alhamdulillah. Okay. So this slide is just a bit of a recap, and it's adding a bit more concepts. First of all, we're saying what factors are affecting the smooth muscle contraction. Only MLCP controls the calcium sensitivity. Yes, MLCP controls the calcium sensitivity of the smooth muscle cell. Only MLCP. Now we're going to be talking about factors that affect smooth muscle contraction. So, we're basically doing a recap of what we discussed and adding a few more factors. First things first, MLCK and MLCP are obviously like regulating the smooth muscle contraction. If you have more MLCK, you will have more contraction. If you have more MLCP, you will have more relaxation or less contraction. Same thing. Furthermore, if you increase the IP3, because remember I said IP3 receptor is what causes the sarcoplasm, the calcium to release from the sarcoplasm. So if you increase IP3 and MLCK, then you're going to have even more smooth muscle contraction. On the other hand, if you increase MLCP, then you'll have more smooth muscle relaxation. What about phosphorylation? phosphorylation acts in a bit of a weird way here, and he didn't go into the details of these two pathways, so just memorize them. Number one, when you phosphorylate something, when MLCK gets phosphorylated by CAMP, so just memorize the CAMP. If it's acting on MLCK, it's going to decrease its activity. And what does that mean? You will have decreased contraction. And I'm sure there's a very logical pathway behind this, but number one, you do not have the time for that. Number two, he did not go into those details. So just memorize it. CAMP. If it increases, then it's going to decrease the activity of MLCK, which means decrease contraction and more smooth muscle relaxation. On the other hand, you have something called Rho kinase. You do not need to go into the details of what a Rho kinase is, but you have Rho kinase. If Rho kinase is phosphorylated, if it's increased, then you're going to decrease the MLCP, and that means you're going to have more contraction or less relaxation, okay? So these two bullet points, you kind of have to memorize them. It's taking a while for the screen to load. Okay, these two bullet points, you just have to memorize them. And then he went on to talk about hormonal influence, so how different hormones can cause relaxation and contraction. What are the hormones? Over here are your hormones. Okay, you see this side of the slide? You're also going to have to memorize them. This one is for contraction. These four are for contraction. These four are for relaxation. And how exactly does it cause contraction and relaxation? These four will go and act on the IP3 receptor and cause more calcium to release, and then that's going to cause more contractions. Whereas these two are going to go and increase the CAMP, and as I said, CAMP will inactivate or inhibit the MLCK, which means less contraction. Is that clear to everyone? Because I think you just have to memorize them. Okay, great. And then we have the nitric oxide. Nitric oxide will you just need to remember this. Nitric oxide will act on the MLCP and cause relaxation. Can you repeat briefly? Yes, I can. Basically, what I was saying over here on this side of the screen, he put a box, and then he put a bunch of different hormones. What you need to know about these hormones is that these four will cause contraction. How do they cause contraction? These four will go and act on IP3. What did we say about IP3? We said it causes calcium release. So when they go and increase the effect of IP3, you're going to have even more calcium, and now you'll have more contraction. So these four will cause contraction. But then you have these two, adenosine and beta adrenergic receptors. What they're going to do is they're going to go and increase the CAMP. And what did I say about the CAMP? I said CAMP will go and decrease the activity of MLCK, which means you will have less contractions, which means you will have relaxation. So these two relax, these two contract, these four contract. And then finally, my last point was nitric oxide. Nitric oxide will go and act on MLCP by increasing its activity, which means you will have relaxation. So you just kind of need to memorize the effects of all of these individual factors. And we're reaching the last concept, okay? Basically, we're talking about a latch state, and to explain it very simply, wait, first of all, I do need to bring your attention to this. Phasic contractions are usually triggered by voltage-gated calcium channels. And you memorize this in case he brings it as an MCQ. Tonic contractions are usually done by ligand-gated calcium channels, okay? So remember how we said there's two types of contractions, phasic and tonic? Yeah, just memorize what channel causes what type of contraction. Now, what is this latch state that we were talking about? Very briefly, sometimes you do not have a lot of ATP in your system. And we said ATP is needed to, let me just see the slide. We said that the ATP is needed to allow for cross-bridge cycling to occur. Because if you go over here, if you stay stuck in the state, if you stay stuck in the rigor state, you're just going to stay contracted and you're not going to do anything. But what happens in cross-bridge cycling is that you need ATP so that this cycle can go over and over again and continue the movement. But sometimes you do not have enough ATP, and you need these contractions to last longer, so you can still be able to contract even if you don't have enough ATP. So what's going to happen is that you're going to enter a latch state where you basically just stay in this rigor state for a while, and you hold the contraction for a while longer. And why does this happen? Because you do not have enough ATP in your system. That's all you need to know about that slide, honestly. Do you guys have any questions? Oh wait, not here. This is our final concept. So if you guys need me to repeat anything from the past two lectures, just let me know. Okay, since I didn't get any questions, this is our final question before you guys are free to leave. Yes, absolutely. See, thank you guys. And alhamdulillah, that wraps it up for our lecture for today. Thank you guys for your patience and for sticking with me throughout this lecture. Inshallah, you do so well on your exam. And yeah, you can leave the meeting so you guys can have a break before your next lecture.